Enzyme Tales: How Beta-Glucoside Split Lactose to Free Glucose and Galactose. This is Henry. He just turned 1 year old today and is dancing to celebrate. He decides to dance all night long. But soon enough, Henry became upset. He is tired. His energy supply has been depleted and he could not dance anymore. Henry drinks his bottle of milk, which contains tons of lactose that can be broken down to form glucose and galactose and used for energy. And after a while, Henry falls asleep. He begins to dream about the enzyme Beta-Glucosidase and how he split the substrate lactose into the two monosaccharides, glucose and galactose. So let's take this opportunity to see what is happening inside Henry's dream, and it all starts within the small intestines. Within the brush border of the small intestines, microvilli lives an enzyme called Beta-Glucosidase. This enzyme will assist in digesting Henry's milk and breaking down the substrate lactose, which are very grumpy due to not being able to pass through the absorption cell of the small intestine. If we take a good look at the grumpy substrate lactose, we can see how it has hydroxyl groups on each of the aromatic rings, but the hydroxyl groups contributes to the structural stability during the breakdown of lactose. It is important to note here that glucose is on the right and galactose is on the left, but they're in disguise. As well, the glycosidic bond is the oxygen bridge that connects glucose and galactose. And if we take a good look at the happy enzyme Beta-Glucosidase, we can see that within its active site there are glucamic acid residues called Glu537 and Glu461, which is a carboxyl functional group. Glu537 helps with forming a covalent bond with galactose during galactosization, which we'll get into in a moment, while Glu461 functions as both a proton donor during galactosation and a base during degalactosylation, assisting in activating water and breaking the covalent bond. So the reaction between the enzyme beta-glucoside and substrate lactose will occur in two steps: enzyme galactosation and enzyme degalactosylation. So step 1 involves galactose becoming covalently linked to beta-glucosidase through the nucleophilic movement of Glu537. This starts by the proton donation by Glu461. This step ends when glucose breaks off from lactose. Step 2, this step involves the breakage of the covalent bond between galactose and Glu537 with the help of a water molecule acting as the acceptor. This step regenerates beta-glucosides and releases galactose. The hydroxyl groups of lactose binds to the active site of beta-galactosidase, positioning it for galactosylation. Upon binding, the enzyme substrate complex is formed. Lactose is shown not directly connected to beta-galactosidase, and this is to better show the chemical action occurring between them. The nucleophile Glu537 performs the nucleophilic attack on the carbon 1 of galactose and donates an electron from its oxygen. Glu461 donates a proton during the nucleophilic attack. This will form a covalent bond between galactose in the next steps. Glu537 and Glu461 stabilize the transition state. The transition state is formed just before the glycosidic bond fully breaks and a new covalent bond forms between galactose and Glu537. After Glu461 donates a proton to its glycosidic oxygen of lactose. So this weakens the bond between galactose and glucose. The nucleophile Glu537 attacks the endomeric so the carbon 1 of galactose, forming a covalent bond between galactose and Glu537 and releasing glucose from its active site. So this lonely aromatic ring is actually happy glucose. So this is the happy glucose molecule that was in disguise. Glucose can now get transported through the absorber cell of the small intestine by use of a glucose transporter, GLUT, by facultative diffusion. Glucose then enters Henry's bloodstream, where it can be transported to different organs that possess mitochondria that can convert glucose into ATP through cellular respiration. But this amazing reaction isn't done yet. The hydroxyl group of the half-lactose, galactose in disguise, binds to the active site of Beta-Glucosidase, positioning it for degalactosylation. This is the start of the hydrolysis reaction. A water molecule, or an OH attached to an acceptor, enters the active site of the enzyme. Water plays an important role in breaking the bond between the beta-glucosidase and galactose. Glu461 acts now as the base and assists the water in attacking the anomeric carbon, so carbon 1, of galactose. This begins when Glu461 donates a proton and functions as the base during the nucleophilic attacks. Both Glu461 and Glu537 help stabilize the transition state of the reaction, enabling efficient cleavage of the glycosilic bond between galactose and the water molecule. The Glu461 carbonoxyl acts as the base catalyst, extracting a proton from the water molecule and forming a nucleophilic hydroxide ion. The hydroxide ion now performs the nucleophilic attack on the C1 carbon of galactose that is covalently bonded to Glu537. Galactose is released, allowing the enzyme Beta-Glucosidase to regenerate for when the next lactose falls within its active site. So, surprise! This lonely aromatic ring is actually happy galactose molecule in disguise. Once galactose is free, it is absorbed through the walls of the small intestine and enters the bloodstream. Galactose is then transported to the liver and is metabolized into glycogen, where it is saved for future energy use. Beta-Glucosidase is now free to bind to another lactose to begin the reaction over again. Henry finally wakes up. He feels refreshed after dreaming about this amazing enzyme substrate reaction and is ready to dance all night again.